Good morning, everybody. I hope I am audible to one and all.
Yes, you are audible.
Please go ahead.
Thank you. So I welcome all the participants. Today we have one very interesting speaker with us, Dr. Manuel Thomas from Kerala. And I am really happy that in this PhD webinar series, we are doing the series with our ninth speaker today. And we want to continue it. So in the interest of time, I would like to introduce our today's speaker, Dr. Manuel Thomas. Dr. Thomas is currently working as research consultant, Unibiosis Biotech Research Labs, Cochin, Kerala. Dr. Thomas is life member, Society for Indian Human and Animal Mycologists. He is a founding member of Research India Foundation. Dr. Thomas is a reviewer for Asian Journal of Infectious Disease. He also is reviewer for Journal of Pharmaceutical Research International. He is reviewer for Uttar Pradesh Journal of Zoology, and he is also member of editorial board AS Veterinary Sciences. Dr. Sir has prepared a checklist of microfungi in Kerala in 2011. He is member in research team Nate Singh Credit, which was published in United Nations Conferences Parties 26, COP 26, which happened at Glasgow in 2021. He has also filed one patent regarding his research work. Other than that, Dr. Sir has published more than 30 national and international peer-reviewed research papers. He has participated in more than 10 national and international seminars and workshops. And Dr. Sir is a reviewer in international conferences on human and technology, a holistic symbolic approach to sustainable development organized by MES College, Marampally, Aluva, Kerala, India. He has also received one Outstanding Researcher Award in 2022. Now I would like to welcome our today's speaker, Dr. Manuel Thomas. Sir, please.
Am I audible?
Loud and clear.
Okay. Thank you.
Thank you.
First of all, I would to thank the organizers, especially Society for Microbiology Society, for opening a platform, such a wonderful platform for sharing the research findings of budding scholars. And I am deeply congratulating all the persons behind this wonderful program. Thank you all. Now, the topic of my research is molecular sequencing and efficacy of plant extracts on fungi prevailing among water rats, that is water norvegicus, inhabiting Vembanad wetland agro-ecosystem in Kerala, under the supervision and guidance of Dr. M. Thangavel, who is now working as Professor and Dean, School of Life Sciences, Nehru Arts and Science College, Coimbatore. So we all know that rodents, they are the largest order of mammals with more than 2,000 species. And among the living mammals, over 40% belong to this order Rodentia. And their success is probably due to their small size, short breeding cycle, and ability to grow and eat a wide variety of foods. Besides this, they are the principal vertebrate pests of crops and crop products. They are inflicting heavy loss to our crops. More than that, they inflict damage and loss to field crops. Their stored food has also significant impingement on public health too, because the rat excreta they are excreting, which got contaminated in the food. So they are the most nauseous vertebrate terrier animals. According to some studies, they transmit more than 260 diseases across the world. And the most interesting thing, majority of the emerging zoonotic pathogens are harbored by the various rodent species. And among all the pathogens, the rodent-borne fungal pathogens, they are the least studied. Even though the chances of transmission are very high, even in tropical environment, where the contacts are unavoidable. So the recent complex manifestations associated with the immunocompromised individuals also paves the way for getting more relevance for rodent harbored fungi. So there are two routes, that is direct and indirect, for the chances of routes of transmission between rodents and to humans. That is the direct and the indirect one, like the plague, rodent to arthropod vector and then them to humans. And the second one, the rodent to livestock and food products to humans, and rodent excreta like the leptospirosis excreted to the water and then to humans. And the most important thing is due to the climate change and fluctuating weather conditions, the rodent populations are highly affected and they are increasing their population here and there. And Reuters in 2003 termed the rodent-borne diseases as human health time bomb. And Began in 2003 in his article in the famous book Rats, Mice and People, he called the rodent-borne diseases as sleeping giants. And majority of the diseases of unknown origin, the researchers are suspecting rodents as the reservoir. So the rodent-borne diseases can be bacterial like leptospirosis, plague, rickettsiosis, whether it is parasitic like leishmania, viral like hantavirus and arenavirus, or fungal like penicillosis, marmorrhea, immunosiosis, etc. And coming to fungi, mycotic diseases, they are the most neglected group among the pathogens. Even the healthcare providers will no need to report it, as there is no immediate public health action is required. So the major objectives of the study: to isolate and identify prevalent fungal pathogens carried by a Rattus norvegicus, the water rat, in Vembanad wetland agro ecosystem. Also to find out mycological quality of water media in the ecosystem. Also to study the prevalence of soil inhabiting fungi in the wetland agro ecosystem. And certain plants in the Zingiberaceae family was which are very commonly used by the traditional herbal practitioners in the locality was spotted out.
मैं चार्ज पे यहीं लगा देता हूँ, आप फोन भी यहीं लगा दो.
Excuse me, everybody, please keep yourself muted.
Okay. Okay. So these plants are selected and tested their activity against the prevalent isolates isolated from the rodents. So this is sample location. It is in Kerala. Kottayam is the district, and where Kallara is the name there. And the thing is, about 350 acres of land are surrounded by water, and where they are cropping oil palm. The farmers are cropping their oil palm. So the locally it is termed as Nindur Kunnan oil palm plantation. So the thing is, the farmers are due to heavy crop loss. The farmers are setting the traps in every evening. About 500 traps are setting every day, and in the morning the trapped rats are collected and killed by the farmers, and that killed rats were used for my study. The rats collected and killed by the farmers were weighed, morphometrically measured, sexed, and taxonomically identified. The samples were collected according to the protocol developed by Hiberto et al. In 2011. The dorsal hair, ventral hair, tail scrapings, forenail, high-nail, whisker, and skin lesions, if present, were collected for further mycological analysis. All the samples were inoculated into SDA with antibiotics and kept at 25 degrees for 10 to 15 days. And after that, in parallel to that, the soil samples collected were serially diluted, spread plated, and isolated and identified, and also a hair baiting technique as recommended by Van Brussigam in 1952 was also used for getting more isolates of keratinophilic in nature. The water samples were spread plated and identified. For identification, a Scotch tape method as reported by Davy et al. In 1996 was adopted. Coming to identification, the cultural characteristics like color of the colony, reverse pigmentation, colony texture, topography, colony margin, pattern, exudate production, rings of growth, And the microscopic characters like the properties of hyphae, presence or absence of chlamydospores of different types, arthrospore, conidiophore, conidia, and physiological characteristics was used for identifying the fungi. And a battery of media was also used for identification, for easy identification by the slow growers of fungi, like the Sabrodextrose agar, Protectodextrose agar, etc. And the slide culture and water culture was also employed for in certain cases for identification. So this is a photograph showing the results of Van Brussigam hair baiting technique, where the samples were collected from the agro ecosystem. The hairs are inoculated hairs are fully attacked by the fungi, keratinophilic fungi. And the most prevalent isolates from the rats, soil, and also from the water was subjected to molecular analysis, that is blast analysis. And I have already mentioned about the antifungal bioassay of these are the plants selected as per the advices of the local traditional practitioners there. The Singiber professionale, Alpinia galanga, Singiber sorumbet, and Kempferia galanga were the plants collected. These are dried under shade and crushed with mortar and pestle, and 20 grams of the powdered material was soaked in 200 ml methanol for 1 week and filtered and evaporated to obtain the residue. And with this residue, the experiment was conducted. Agar well diffusion method. These are photographs of the plants used for the study. The pure cultures of Absidia corymbifera, Aspergillus niger, Chrysosporium species, Curvularia geniculata, Fusarium semitectum, Phyllomyces varioshi, Syncyplastum racemosum, and Trichophyton rubrum were used for the study, which are the most prevalent isolates from rat samples. A total of 190 water rats were trapped and killed, which are trapped and killed by the farmers during the period September 2013 to August 2016 was used for fungal pathogens. Altogether, 88 species of fungi were identified from the water rats, followed by 74 from soil and 67 from water samples. So the bar diagram showing the prevalence of fungal pathogens from water rat, soil, and water in the wetland agro ecosystem in Vembanad in Kerala. So we can see that the genus Aspergillus is the most common genus represented in the study from the water rats, followed by Fusarium, Penicillium, and Trichophyton. In the sex-wise analysis, there is not much difference between the career status of male and female, and about more than 40 were present in both males and females among the isolates. So probably ventral hair showed more fungal carriage, followed by dorsal hair and tail scrapings. This is a genus-wise representation of, like the rats also here, Aspergillus, Fusarium, and Penicillium, and Trichophyton are the prominent isolates, represented genera. But in the case of water, the most common genera represented are Aspergillus, Fusarium, and Penicillium. So the seasonal comparison of isolates in connection with the seasons. The southwest monsoon in the study, southwest monsoon is the most favorable for fungal presence in the study. So comparison of CFU month-wise. So we can see that in the case of soil, there is a sharp increase during the monsoon, that is May-June period. And in the case of water, the rate is more or less steady in the study. These are the photographs. I have already mentioned the characters employed for identification of different species. This texture, whether it is cottony, mucoid, velvety, powdery, granular, or glabrous. Coming to topography, whether it is vergose, flat, cerebriform, crateriform, or ambonite. Coming to the other features, like it is feathery, irregular, restricted, spreading, or exudate production. In the case of Aspergillus and Penicillium, the employing exudate production, inducing exudate production is quite easy for identifying the unidentified species. The rings of growth, various color combinations, and agar collecting property also employed for identifying certain species. These are the photographs of the major isolates: Absidia, Acrimonium. About more than 15 species of Aspergillus was obtained in the study. These are photographs. Sorry. So water rats in the water science, they are represented by 15 species of Aspergillus, followed by Fusarium, 9 genus, and Trichophyton, and 8 genus, and Penicillium, 7 genus. So from the soil samples, the major genera includes Aspergillus, that is 11 species, followed by Fusarium with 9, and Penicillium and Trichophyton 7 each, followed by Microsporum and Mucor, 4 species each. In the case of water samples, here also Aspergillus was more common by 11 species, followed by Penicillium 9 and Fusarium 5. We all know that aspergillosis and related diseases are emerging and very common nowadays, with the aspergilloma or invasive aspergillosis, and even the World Health Organization has started celebrating World Aspergillosis Day on February 1st since 2018 to make aware the people about the emerging situations of aspergillosis. And this is one of the during the study, the lesions I have already mentioned about the lesion samples. In this, right, the lesion obtained and the sample was collected and cultured and identified as cytalidin and dimidiatum. It is inflicting superficial skin diseases and onychomycosis in humans. The characteristic is very nasty smell and baroconidia, arthroconidia. So for molecular analysis, the rat samples, the more 72.22% was Trichoderma hametum's prevalence, followed by Aspergillus niger and Aspergillus flavus. The soil samples also the same species, and all these 9 cultures were subjected to blast analysis and confirmed the strain circulating in both rat, water, and soil in the ecosystem. Trichoderma hametum, the blast analysis of Trichoderma hametum, Aspergillus flavus, it is during the blast analysis, it is identified as Aspergillus nomeus, which is a strain of the Aspergillus flavus complex. And next is Aspergillus niger. So coming to antifungal activity studies, Alpinia galanga yielded was more sensitive against all the 10 fungi tested, followed by Zingiber officinale. So this is a photograph showing the sensitivity pattern, the zone of inhibition of the tested fungi with the various extracts. So water rats are carrying a variety of fungal pathogens with zoonotic potential in a wetland agro ecosystem. Seasonal variation and substrate preference by fungi are observed. Sequencing studies have revealed that same strains are circulating in the rat soil water nexus. The presence of emerging pathogens in a reservoir which is co-evolved with man is quite detrimental in conditions like in a tropical environment and also in an agro ecosystem. The antifungal potential of locally available medicinal plants can be tapped to combat emerging fungal diseases. And the growing population of immunocompromised individuals makes the situation more complex and vulnerable. The rat soil water nexus in fungal ecology and transmission are complex with multifaceted public health implications to the interacting community. The typical ecology prevailing in Kerala favors easier transmission of fungal pathogens to humans, especially in a wetland agro-ecosystem, where contacts between rats and humans are inevitable. The findings raised higher public health threat. And we all know that rodents furnish a link between wildlife and humans, displaying humans and his livestock to zoonotic diseases circulating in natural ecosystems. So, the zoonotic risks associated with rats in agro-ecosystem cannot be ignored in the wake of emerging zoonotic and fungal diseases. My acknowledgement goes to my guide supervisor, Dr. M. Tangavel, Department of Microbiology, where part of the work was conducted at Srinaranaguru College, Coimbatore, Department of Microbial Biotechnology, Bharatiyar University, Coimbatore, Department of Microbiology, College of Veterinary and Animal Sciences, Mannuthi, and Department of Medical Microbiology, School of Medical Education, Mahatma Gandhi University, Kottayam, and Rajiv Gandhi Center for Biotechnology, Trivandrum, where the sequence analysis was done, and the CTAB, where majority of the study was conducted, Consortium Training Academy for Biosciences, Kottayam, and Nizam Institute of Medical Sciences, where I got a workshop of 13 days, and Society for Indian Animal and Human and Animal Mycology, SIHAM. Thank you. Thank you for providing me an opportunity in this platform. Thank you all.
Thank you, sir. Thank you very much. It was an interesting lecture. Now we are open for question and answers. So everybody, whoever has questions, kindly unmute yourself and you may ask. And please, everybody, you can also use chat box for the question answers. I think Prakash sir has some question.
Thank you, Dr. Thomas. Can you hear me?
Yeah.
Wonderful. First of all, I would like to congratulate you for such a nice work. Actually, just I wanted to inform the August gathering that beside heading the Microbiology and Fermentation Technology Department, I am actually with the additional charge of food protectant and infestation control, where controlling the rat in a warehouses, godowns, store grain, which actually consumes the large amount of the store grain. I hope Dr. Thomas will agree with me that rat actually consumes 25 to 30 grams of the grains per day, and very high, the populations which we actually see, and actually a major destroyer of the most of the store grains. Coming to the topic which he has studied with reference to studying the fungal diversity, whether you have seen any newer novel species of a fungi in a GI tract of rat. That is what actually I wanted to know. One thing, another thing was how you know that these were the pathogenic fungi. I hope I am clear.
Actually, I have not done any study with the gastrointestinal tract. I have taken only samples from hair and nail and whisker only.
Okay. Yes. But how you know that they were pathogenic fungi?
No, I have not tested the pathogenicity aspect, but interpret with the available literature.
Exactly. And then the soil and water sample you have taken only for comparison, but because your topic as such is for the rat, right?
Sorry, sir.
In your topic, the major emphasis was studying the fungal pathogens in rat. Not soil and water, but soil and water, the fungi obtained from soil and water, you use for comparison.
A comparison, and also I have collected samples from all the three samples, that is rat, soil and water from an ecosystem, wetland ecosystem.
Exactly. Did you find any newer novel species of fungi?
No, not yet.
But it is actually a very good pictorial representation which you have shown. The colony morphology as well as the SE pictures was really wonderful. Okay. So congratulations once again. Nice work. Thank you.
Thank you. And I have struggled so many. I have taken about 12 years to complete the work because nobody is accepting, so nobody is guiding to identify, and at last I have contacted Dr. Randava from Vallabhai Patel Test Institute, and he directed me to contact SIHAM, where Nizam Institute is conducting a workshop of 13 days. And there, actually, I got proper guidance in identifying and various other things.
Oh, very good, very good.
Thank you, sir, for your inspiring comment.
Thank you.
Indeed, it is very wonderful work, sir. There is absolutely no doubt. And there is one more question in the chat box. Milinji is asking, how you selected plants for antifungal activity?
How you selected? I have contacted the traditional healers, traditional medical practitioners, herbal practitioners in the locality, and I have selected the plants which the plants are used by them for healing various skin diseases for people in that area.
Yeah, just Jay, another actually important point just I wanted to tell is that we do not have much antifungal substances of a microbial origin or from the plant sources. Whatever we have, the antibiotics are the chemically synthesized. So this is actually another unexplored area. One can actually look that the antifungal substances either from the plant or from the microbial sources is actually very important. And then we see even in the food industry, the fungal spoilage is a major one where we use the chemical preservatives right now also. And then you know that most of the chemical preservatives, they are under now banned, and then they cannot be used in a higher concentration because of their carcinogenic and mutagenic effect. So I think looking those plant metabolites or this plant extract against the fungi, even in the food industry also has a lot of importance, in other words. Yes. Yeah, go ahead.
Yes, sir. Dr. Upasni is asking, have you deposited unique isolates in the culture collection centers?
No, not yet.
All right. Vivek, sir,
Please do.
welcome. Please unmute yourself.
Sir, I am keeping the pure cultures, and definitely I will deposit it.
Yes, please do so.
Okay.
Thank you. Thank you. Because I have seen people losing cultures, and I also lost some cultures because of negligence from the lab maintenance.
Oh, okay.
Right. So it's always safe to have cultures deposited, and it's quite fast. And I have done that. So I think NCCS provides good facilities for that.
Yeah.
One more question, which is sequencing method you have used? Actually, I depend on RGCB, Rajiv Gandhi Center for Biotechnology. I have given the cultures and they have done it. I am also not well versed with the molecular type work. So I have done blast only for confirming the identity only, the strain side level.
All right, sir. Do we have any more questions? Anybody?
We just wanted to know what is the now present activities of Dr. Thomas, which he is pursuing in some company.
I am
also inspiring for the several PhD graduate from our society.
Okay. The first thing is I am a research consultant at Unibi's Biotech Research Labs in Cochin.
Okay.
And also I am a mycologist in a pet hospital, Dr. Su in Ernakulam.
And what is the work you do there? Means what is what they are looking?
According to the doctor's estimates, assumptions, about 40% of the pets are have fungal infection. But normally,
exactly, that is what actually I was trying to tell. Not only for the pet or the veterinary animals, but even our skin infection, and we do not have actually the potent medicines. And similarly, in the food industry or also in the food system, we have the microbial preservative for a bacteria. Okay. But not for the fungi, right? And this actually, again, we see the problem even in the plant tissue culture. In
Yeah.
plant tissue culture also, avoiding the fungal pathogen is a challenge, in other words. Yes.
And there I am serving as a mycologist for they are taking samples and culturing for identification.
Oh, that's great.
We also have a question in the chat box.
Which solvent is best? Which solvent is best plant extract? It depends on the type of work you are designing. That is organic or inorganic solvent. It depends on the objective of the work of the isolate. I have used alcohol because it is only a screening work where we can get unique isolates for antimicrobial activity. I am working on Tridax procumbens plant extract, which is antifungal wound healing. Deepika Kale. MTCC is providing unique cultures, microbial type culture collection. And Agarkar Research Institute are also providing such services in Pune.
All right, thank you, sir. Sir, I have one question myself, if I may. Would you like to share what kind of challenges you faced in your PhD and how did you come up with that? I think that will inspire everybody.
The first thing is actually I have started my research with the hantavirus, and that time in US, I think it is in 2004-06 period, four corners outbreak and something like that. And I have searched the literature and discussed with my guide and connected with one professor from Argentina, Dr. Noema Pini, and she offered kits. But the thing is that I need immunofluorescence microscope and not able to get it. So I have dropped it. And for that purpose, I have collected rat excreta from the field for 100 or 120 extracts are there. Actually, after more than 6 months, I am planning to clean my cupboard. And there I noticed different colors in the excreta pellets. Some is green, some is black, some is olive green. And there I have started to culture fungi. And I don't know anything about fungi. So there I conducted School of Medical Education under Mahatma Gandhi University, where there is medical microbiology department. And they guided me, and they given me a book of Fisher and Cook of Fundamentals of Medical Mycology, and take a Xerox of that book and read it within 1 or 2 weeks. And within 1 month, they are unable to support me because I am culturing everything, soil, everything, even my hair also I have cultured. And there I started my journey with cultures. I have gone to Coimbatore, GRD College, Veterinary Sciences University, there even TNAU in Coimbatore, Madras Christian College, everywhere, Madras University, but nobody has interested to support me. And there I have contacted, I have already mentioned, with Dr. Randava, and he guided me to the right place. So it is quite risky, and all people are advising me, it is quite risky. But I don't know, I have not even get a ringworm for this time, even at this time. And yesterday also I have made culture, and even at today I am lucky that I have not contacted anything during my past 16 years of play with fun guys.
Great to know, sir. Great to know. Sonali is asking how to select guide. I think it's a debatable question, but whatever you might want to share, sir.
Roughly speaking, I have even cancelled my first guide and changed with the another one because I have no way to cooperate with that man. So it is the most crucial thing in your PhD.
Right, sir, right. And Prakash sir is mentioning how is nutritional requirement of fungi obtained from rat?
Actually, I have not done.
Basically, Dr. Thomas, whether you were finding a difficulty to grow them because maybe soil and then water fungi, it is okay. But I think from the one which grows in a not bad body.
Yeah, that is why I have used more than 10 media for culture. And even I have used my blood, 1 drop of blood for 500 ml for added nutrients.
Oh, that's challenging.
Yeah. And also got, that is why I have got that interesting photographs. I am changing the nutrient composition every time. So if any culture is growing very slowly, then I will subculture with and added with some nutrients.
Oh, okay. So you have optimized a lot of such laboratory media, you mean?
Yeah.
What are the other nutrients you added? Just like what you said, supplement you incorporated in your regular PDA or DFO, whatever the media you use for the fungi cultivation.
Blood and even serum.
What is that another one?
Serum.
Oh, serum. Okay.
After centrifugation. After serum. And then adding in SDA, we are adding I am added potato also, a tinge of potato.
Wonderful.
Like that.
All right, sir.
Oh, thank you.
Deshmukh sir, are you there?
Yes.
Oh, all right, sir. We are very thankful to this platform.
Pardon?
I am telling that we are very thankful that you are providing us this platform.
So I'm more delighted today. The reason is that the young generation of India is doing extensive research work, and they are inspiring other research workers of India. Today's lecture is recorded, and very soon, within 5 minutes, I will circulate in all groups. And I'm sure, because I can see the number of viewers on the YouTube, and many students enjoy this lecture later on. So thanks to everyone, and particularly senior faculty of India attending today and inspiring the young faculty of India. So that is a very good discussion today. Full to everyone. Thank you all.
Thank you, Deshmukh sir. Thank you very much.
Thank you all for inviting and allow me to share my research findings and also experiences with you people.
Thank you so much, sir. Thank you very much. Thank you, everybody, for being there.
Thank you, everyone. Thank you, Dr. Thomas, for sharing your PhD work. Thank you, everyone.
Thank you.
Thomas, sir, just one last request. Can you type your email in the chat box?
Yeah, sure.
Yeah.
It is also interesting. drfungus0007@gmail.com.
Oh, very nice.
drfungus0007@gmail.com.
Okay, so if anybody has any further questions, we can follow through that. And yeah, have a nice weekend, everybody.
Okay, thank you. Thank you all.
Thank you. Bye.